What if a cancer drug could hitch a ride straight into a tumor's cell nucleus, killing malignant cells while sparing healthy tissue? Welcome to Industry Ignited, the podcast where we spotlight leaders who are building the future of industry and healthcare. I'm your host, Dr. Leeanne Aguilar. Today, we're joined by Paul Jaminet, founder and president of Angiex and author of Perfect Health Diet. Paul's journey runs from MIT physics and Harvard astrophysics to software to launching Angiex, whose lead therapy, AGX 101, a TM4SF1 directed nuclear delivered antibody drug conjugate, is now in phase 1 clinical trials. Paul, welcome to the show.
Thank you, Leanne. It's great to be on with you.
You started in astrophysics, then helped build software companies, then moved into natural health, and finally oncology. What through line connects those chapters?
Well, I guess the common feature is that I've always considered myself a scientist and almost my whole career an entrepreneur as well. And I would say that the two of those actually have more similarities than most people realize. So at least the kind of scientific work I did, it was mainly pulling together distinct ideas, concepts, evidence, doing experiments to generate new evidence, and then pulling everything together to make a coherent whole. And entrepreneurship, I feel, is very much the same, except with people instead of ideas. So you're pulling together a group of people to form a new network that produces something new that has, just like you want a scientific theory to be coherent and but also to be beautiful, to be to give you insight. And in entrepreneurship, you're trying to pull together a group of people to create something new and beautiful, something that does good work together. And I think, a lot of the thinking and work that I've had to do in both areas is much the same. It's just the difference between ideas and people, but they get pulled together in much the same way and, much the same kind of thought. So I would say if there is a through line that connects everything, it's those. My career has taken a number of turns, as you mentioned, and so when I was young, I didn't really know what I wanted to do with my life, but I loved science, and so I figured I'd start my career as a scientist, and so I stayed in academia, maybe too long, but it was it was kind of the easiest path forward while I was figuring out what I wanted to do with my life. And then I was successful in it. So I did my undergraduate work at MIT, as you mentioned, got a PhD at Berkeley. And then I worked in astrophysics at Harvard for 4 years. And I could have stayed in academia, but I decided I didn't want to spend my career in that, and kind of the opposite. Academia was too stable for me, and I wanted something a little more changeable and exciting. And at that time, the internet was invented, so the opposite extreme in excitement seemed to be starting a software company. So that's what I had the good fortune to find some partners and be able to do that, and that was a lot of fun. So I've considered myself an entrepreneur ever since. So we started that in 1996, sold it in 2001. With the same founder, started another company, but they wanted to be in California, so that company moved to Silicon Valley, and I wanted to stay with my wife, who was a biomedical scientist in the Harvard system, so I stayed in Boston. And then I got interested in learning how to be healthy, and once I learned how to be healthy, I felt I had a duty to share it, and so I started a natural health business with a blog, a book, and a health retreat. And that was also a lot of fun, and I met a lot of great people doing that, and we had terrific health results. But meanwhile, I told my wife if she discovered a way to cure cancer, then I'd help start a company to make it real, and she did. So we had to start
Wow.
Angiex in 2015, and that has been another exciting journey.
Yeah.
So we got off to a good start with seed funding from Peter Thiel. We were able to make a great drug. There's been some ups and downs, especially in the COVID era, but we're in the clinic and the drug is doing very well. So,
That is, yeah, very exciting. Lots of twists and turns, and it sounds like quite a journey. What moment, though, you mentioned your interest in health, and I know you mentioned the retreats, and you still have a website out there that's active. Are you still doing that in any capacity?
well, I'd like to start again because we had so many great readers. I'd love to get back in touch with them, and so I hope to resume some blogging. And now that our drug is in the clinic doing well, I think the world needs to hear about it. So I'm trying to carve out some time to resume blogging, and both on the Perfect Health Diet site. So the site is perfecthealthdiet.com, but also starting a blog on the Angiex website. And
Was there a defining moment in which you decided, like, we have to start a cancer company? Was it just that your wife had gotten far enough along in the journey that you saw it was time to switch over to that focus?
yeah, so I had kind of a premonition very early on, first of all, that she would discover a way to help a lot of cancer patients, but then also that I was familiar with academia, so I know what its limits are as far as what you can do, and you definitely can't develop a drug. It takes typically $300 million to bring a drug to approval, and I've never heard of anyone in academia being given $300 million to do anything. So I was following along her work, and she's not a native English speaker, so I would help her with her papers and her grant application. So I was intimately familiar with all of her work and following along with it. And, so I could see what was going on, and I knew when the time came when it was the right time to launch a company.
Okay. And building Angiex with your wife, I mean, she is one of the scientific founders. She's the one behind all of the research, right? Um, what have you learned about pairing world-class biology with entrepreneurial execution?
Well, you need both in order to make a great drug, and yeah, they're both essential. And uh, my wife is a magnificent experimental biologist, and very few people could have made the discoveries that she made. But being a great scientific discoverer has its downsides, which one of which is if you're really far out ahead of everybody else, then they may not be able to follow along, and they may not realize the significance of what you've done. And she was in that situation, so she had a difficult time getting grants and progressing her work. So um, uh, and I had always believed that if you have a great opportunity that can help a lot of people and improve a lot of people's lives, then it's better to develop it in a company so that investors can invest and get a share of the fruits of the results, and that motivates them to support your work a little better than if they're just giving grants and getting nothing in return. So it's definitely easier to build something big and expensive in a company than
Okay.
in an academic lab.
Yeah. So you saw like entrepreneurship is more of an opportunity, like more freedom in it, and it
Yeah,
enables
and
you to go
definitely
further.
as the entrepreneur, you can have more control potentially. Although there's always whenever there's more than one person involved, there's always uh, compromises, and you have to make everyone involved in the project better off, and so it's important to find people who share your mission and will support it.
So finding the people that really align with your vision, and yeah, that makes sense. It makes it a lot easier going going
Right?
forward,
Yeah.
especially when it's a long journey like that. When you're developing something that takes, would you say, like 10 years to develop? I mean, it's not
Uh,
a quick
yeah. So in
process.
our case, it'll be close to, be 12 to 15 years from the foundation of the company to the approval of the drug.
Okay.
And that's on top of a prior 13 years of science done in.
Yeah. So a long journey. Yeah, you better like who you're working with, right? Yeah.
Yep, that's for sure.
So looking back, which habits from physics and software most improved your decision making in drug development?
Well, it's certainly very valuable to have the business experience. So I had the, experience in venture-backed entrepreneurship from the internet boom. And, but then I also had the experience of, my own 100% controlled lifestyle business of, the health retreat and the blog. Um, uh, so I had some good experience, and the health retreat turned out to be extremely helpful because, uh, first of all, we helped a lot of people and made a lot of friends and gained credibility with them. So, I would say our diet book is still probably the most scientifically well-grounded diet and health book available, and people respected that, and respected that the advice was helpful, and so we had a kind of a baseline of network of people who believed in us before starting the company, and that was very helpful. So former guests of our health retreat have invested about $20 million into
Wow.
Angiex. And that was very important because whenever times were tough, and it was hard to get money elsewhere, then I could turn to my personal network, and they always came through and support.
No, that makes a lot of sense. Yeah, because you've developed relationships based on trust and really, yeah, gotten to know people, and so they really understand what your uh, passion is and what you're out to accomplish, and they know you, and so they're willing to yeah, put their money where their um,
Yeah.
uh,
So investors typically they have to believe in the idea and the project and the end goal, but they also have to believe in the people and the
yeah.
team. And um, so at least with the people who knew me from my prior work, they had the confidence in the
Hmm.
team, and that was a huge step forward. So
Yeah.
uh, and particularly since I was changing careers into a new industry, so no prior drug development experience. You definitely needed people willing to take a leap of faith on
Right.
me as a person.
Well, that definitely says a lot about the community you built, and the what you were providing, the benefits you were providing, beforehand, and I can see why you would want to kindle that, and start that down in, like a real community. So let's talk about the science behind the drug that you've invented. In plain language, how does a nuclear delivered ADC differ from standard chemotherapy or conventional ADCs, and why target the TM4SF1?
Okay. Well, maybe I'll start with the biology that my wife discovered. So, what she discovered was a very fundamental pathway. Um, and so, if you go back to early embryonic development, so a few days after conception, then you basically have only two cell types in the early embryo. There's epithelial cells, which are stationary, and there's another cell type called mesenchymal cells, which are mobile. They're like the construction engineers of the body, and they can build structures. They're especially good at building structures with tubes and cavities, like blood vessels or the heart. And but they're the one cell type that can move around the body. They can invade other tissue and go into tissue. They can migrate around the body, and so that's very important in embryonic development. That's how a person gets constructed. But in adults, you're already built, so you don't really need a lot of that biology, and it's generally shut off. But cancer turns it back on. And so in the cancer cells, what's called the mesenchymal transition, the transition from the normal stationary state of a completed organ, it transitions backward to the construction state that's found in embryonic development. And the cancer cells become the mesenchymal type cells that can move around, and they can invade tissue, and they can construct new organs. And so they basically, they go a little wild and start constructing new structures, but in a disorganized fashion. And it's those cancer cells that have undergone this mesenchymal transition that can invade and metastasize, and those are the cells that kill you. And it turns out, in addition to tumors, there's one other time when that mesenchymal biology is turned on, and that's when you need to heal wounds, and especially when you need to build new blood vessels in a healing wound. And so when that happens, when you need to build those blood vessels, the endothelial cells undergo a transition, which is very similar to that epithelial to mesenchymal transition. It's called by different names. It's called changing from the quiescent state to an angiogenic state. So angio is Greek for blood vessel and genic for creation. And so it's a state where they can create new blood vessels, but essentially they're turning on that mesenchymal program, the same thing that makes tumor cells malignant. And so what my wife discovered was this very fundamental transport route that collects all of the proteins that need to get to the nucleus to turn on this mesenchymal program and transports them from the cell surface to the nucleus. And so it's taking, it's basically one bottleneck. It's a transport route, but it's the only transport route that can initiate this mesenchymal gene expression program. And it has and the marvelous feature about this from a drug development perspective is there's a tiny little bit of amino acid sticking out, this protein complex that we can make antibodies to, and they'll bind to it. It's exposed to blood on the endothelium, and the antibodies can bind, and they can just hitchhike along this transport route like a hobo hopping on a freight train. And the same transport route is present in tumor cells. And so we made a type of drug, an antibody-drug conjugate, where the antibody is what provides delivery to these cells. It seeks out all the cells that have made this mesenchymal transition, and it hitchhikes along this transport route to the nucleus of those cells. And we put a therapeutic payload on the antibody, which in the case of cancer, we chose a cytotoxin that kills dividing cells. And so it's basically similar to a chemotherapy, but it differs from the standard chemotherapies that it can't go all over the body. It can only poison cells if the antibody has delivered it inside the nucleus. And so we were able to design, first my wife learned about this biology, and we were able to, we saw that it was perfect for antibody-drug conjugate therapy, and we were able to design a drug that exploits it.
Wow.
And so we have a drug where uh, a majority of the injected drug goes to tumors. And it's basically, we're able to minimize the toxicity everywhere off tumor.
Hmm.
So we really have uh, uh, minimal off-tumor effects. In the clinic, we haven't seen any definite off-tumor toxicities, and we're seeing quite a bit of on-tumor activity. So, so we're bringing a benefit to the cancer patients by shrinking their tumors, and we're with no effect off tumor. And
Wow, that's amazing. Now, you've described this transport route, as you called it, that carries cargo from the cell surface to the nucleus. What evidence convinced you it's broadly active in malignant cells and tumor vasculature, but largely absent in healthy adult tissues?
well, that's the magnificent work my wife did. So she discovered this transport route around 2002, and she made it the focus of her career. We started the company in 2015. And, so 13 years of work, and she just did a tremendous amount of work. She made knockout mice that don't have this gene, and they couldn't form blood vessels, so they all die a few days after conception. If they have one allele of the gene, so every gene you have two copies, and she could make mice that had one copy of the gene, and about half of those would die in early embryonic development due to brain hemorrhage. But if they made it past embryonic day 17, then they would be fine. But tumors would only grow 1/10 as fast. So, if cutting the level of the gene in half leads to a tenfold reduction in tumor growth, then it's very important for tumor growth. And now there was another very helpful indicator in that there was one prior drug made against this. It was taken to the clinic by Bristol Myers Squibb in 1990, and they had no idea about the biology. All they knew was that they had found an antibody which homed to tumors, and it was commonly found in tumor cells and in tumors. And they took it to the clinic to see what it would do. And it worked very well if they put a radioactive isotope on the antibody. And so that was strong evidence that if you made a drug with a conjugated cytotoxic payload, then you could have a big impact on tumors without harming the normal vasculature. And and then we were able to do a lot of studies in animals and mouse models. So we studied several dozen mouse tumor models and thousands of mice, and it always worked. And we just explored a lot of designs, and were able to really optimize the design and keep making the drug better and better. So, so I would say, it's an incremental process. You keep making
Right.
progress, and first you have to understand the biology.
Yeah, I was going to say it's first understanding the biology, then, I mean, also looking at previous studies and understanding, what had been done, and then doing a lot of testing,
Yeah.
yeah, for finding that evidence. Now, safety is everything in oncology. What design choices, whether payload masking, release mechanism, dosing cadence, were pivotal to aim for tumor-specific action with minimal off-tumor effects?
Yeah, there were quite a few. So we benefited before we started Angiex. My wife and her collaborator Hal Dvorak formed a 3-year partnership with Pfizer and made a drug. So it wasn't taken to the clinic, but we got the benefit of Pfizer's drug development experience. So they gave us an education in drug development. And they also gave us a baseline drug design that we could improve upon. And that drug design was basically the state-of-the-art drug design for antibody-drug conjugates as of 2012 or so. And that was relatively early in antibody-drug conjugates, but Pfizer at that time, they were trying to be the world leader in antibody-drug conjugates, and they had made a big investment. They had picked up the first approved antibody-drug conjugate, which was called Mylotarg, when they acquired Wyeth a long time ago. And so they had a lot of expertise in this area, and we learned a lot from them. And they made a drug, and we knew we could make a much better drug. So we had this fantastic biology, and their drug just didn't perform that well. So we knew it could be improved upon. And so I knew when we started Angiex that we wanted to go slowly and do careful experimentation and really find a way to make a much better drug than what Pfizer had made. And we were able to do that. So we found 6 very impactful improvements. Overall, we were able to increase the therapeutic index, which is the margin between the dose that gives efficacy and the dose that gives safety. We were able to increase that tenfold. So Pfizer's drug had a very narrow margin, and we have what I believe is the broadest therapeutic margin of any oncology drug in animals, and I think we're proving now that we also have the best therapeutic margin in clinical cancer patients as well.
Yeah. So your patience and perseverance, paid off over time.
That's right. Yeah, and just good science. So you have to do careful experimentation.
Yeah.
There's a lot of mediocre science that's done out there. There's a lot of people with PhDs who can do the same experiment 6 times and get 6 different results.
Yeah.
And so then you don't know which one is right. And there are people who make good academic careers by they do the experiment 6 times, get 6 different results, and pick the result that makes the best story, and then use that to get papers and grants. But it's not necessarily true. And, whereas we had the advantage of outstanding scientists with great integrity who were also great experimentalists and could do very careful experiments, who knew when they'd gotten an answer that was true and when they'd gotten an answer that was false, and could troubleshoot experiments to make sure that they generated a true answer and could do the experiment reproducibly and get the same result every time because they controlled all the relevant variables.
Okay, right.
And, so I would say that was absolutely critical to even being able to form the company in the first place, is to have that foundation of reliable science and be able to proceed with confidence. And then, likewise, as you design a drug, you have to do new experiments to test how the drug works, and you need to make sure you're getting reliable data there. So I would say, the ability to do good experiments is, it's an underappreciated virtue. It's, the skills needed to successfully get a PhD don't necessarily overlap all that well with the skills needed to conduct great experiments. So, so academic success isn't necessarily the same thing as being a great experimentalist. And, so we had the advantage of having, just outstanding scientists. And,
Yeah, no, I think that, getting the PhD is just the first step. That's just, basically educating you on the scientific method, basically, and educating you on the different types of tests. And you get a base level of understanding, but it's not until, you start going out there and really doing the research and the experiments, designing the experiments. And like you said, there's a lot of iterations to determine what a good research even looks like. And because you want that replicability. If it's not replicable, then you don't know if it's good results, really.
Then you haven't done a good experiment
Right.
because you haven't controlled the variables
The right variables,
that you
exactly.
need to
Yeah.
control.
Enough variables or what have you. So if AGX-101 proves out, does the biology suggest a pan-solid tumor opportunity, or are there tumor types or stages where you expect outsized benefit first?
Yeah, so we think we've made a universal therapy for all solid cancers. So our drug has, because it attacks two different comparts of the tumor, it's got multiple mechanisms of action against cancer. So against the tumor vasculature, we can eliminate the tumor blood supply. And that has the potential to shrink every tumor down to the size of a pinhead, which is a size where a cancer really can't harm you. So so by that mechanism, we don't eliminate the cancer entirely, but we make it harmless. And so patients can just be retreated continuously until, without dying from their cancer. And the great thing is because that mechanism is attacking the blood vessels, which are an unmutated host system, there's no known resistance mechanism to the drug. So that's one way that we help cancer patients. Another way is we can kill tumor cells. And we deliver our payload in abundance to every tumor cell that's capable of invasion and metastasis. So we have the potential to kill off all the malignant tumor cells and convert the tumor from malignant to benign, where you have tumors, the surviving tumor cells can proliferate, but they can't move. And so the tumor just grows like a ball in place. And those tumors, they're usually easy for surgeons to resect, or but they don't invade or disrupt tissue. They just so they generally can't kill you unless they're growing in a confined space, like a brain tumor within the skull, and then they can grow and squeeze the brain. But but if they're growing somewhere else and they're not confined, then you just have this growing benign tumor that doesn't harm it. Isn't a threat to kill you. And so that's another way we help cancer patients. And then the third way is that we found that by injuring the tumor vasculature, we point out the tumor to the immune system. So we make the tumor into a wounded organ, and all the rest of your body looks normal, but the tumor looks like a wound. And anytime you have a wound, the immune system goes in and it looks for anything foreign, like germs, and it tries to kill the foreign things. And so that gives a chance for the immune system to recognize the cancer cells as foreign and kill them. And that immune attack is actually the best way to completely cure cancers, or the most likely way to completely cure them, and get rid of every single tumor cell so that the cancer never comes back and you don't need to be retreated.
Right.
So, so we've got three ways to help patients, and they all have broad applicability to just about every solid cancer patient. And, so we have to see in the clinic what the magnitude of the benefit is. But I look at some of these other drugs and other approaches, and they seem almost barbaric by comparison. They do so much harm to the patients.
Yeah.
The indication that will probably go to approval in first, angiosarcoma, it's a very dangerous cancer, and one of the things that are done to treat it, to try to prevent it from metastasizing, because it metastasizes so readily, is they'll just amputate whatever body part has a tumor. And so, you see these patients who have had their legs amputated because they had tumor, tumors in their legs. And so it's just it's heartbreaking, and they still die in a year. So
Wow,
in
yeah.
most cases, and sometimes they get lucky and there were no metastatic tumor cells. But if just one cell has, migrated into the rest of the body, then it will eventually form a new tumor. And you can't keep taking away body parts.
Body parts, no, of course, yeah.
Uh,
Yes. So your technology, your drug will target those cells and
yeah,
yeah,
it'll go through the body and find all those cancer cells and attack them. So.
very promising. So you're in phase one with first in human experience. What early readouts like PK/PD, safety signal matter most for your go or no go decisions and dose optimization strategy?
Yeah. So, well, uh, so our situation is different than a lot of other cancer drugs. So most cancer drugs, they don't work that well. They have a very narrow therapeutic margin. And so the result is that in most patients, they have a negative therapeutic margin. They cause toxicities and harm before they give efficacy. So most cancer companies, they first start by trying to find what's the maximum dose they can dose at that doesn't give, terrible harms to the patient. So that's called the maximum tolerated dose. And then they'll go find a very specific patient population where that's at the extreme end of safety on the good side and the extreme end of efficacy where the cancer is most sensitive to the drug. And they hope in that small population to get efficacy with safety. And so a lot of clinical trials, first they serve a broad population and they just try to find what's the highest dose we can dose at and be tolerated, and then they go try and find the specific patient population in phase two to demonstrate efficacy, and then they just get approved for those specific patients. And often they have biomarker strategies and other strategies to select who are the patients where the drug may work. In our case, because we're almost universal in our mechanisms of action and very safe, potentially, we have the potential to reach efficacy ahead of any safety signals. And so far, it looks like that's the way it's working. So we're now dosing at what we believe is the optimal dose, and so far, at least, we haven't seen any off-tumor adverse events of significant severity. So, so the drug is proving, quite safe. It acts on the tumor and it doesn't do that much off tumor. And, so it just takes a little bit of time to demonstrate efficacy, takes repeated dosing to shrink the tumors down. And you have to follow the patients and, to see that their lifespan is extended and things like that. So it takes some time to demonstrate how well the drug is working. But at least so far, we appear to be safe at what we believe is the optimal dose. And now we just have to establish efficacy and demonstrate to the world that.
Right. Go through the trials, go through the process.
Right.
You've noted the need to chip away versus chemical surgery for certain tumor locations. How are you operationalizing that and like endpoints, imaging cadence, and risk
Yeah.
mitigation?
So that's an important clinical question. So in mice, what we found is we can just keep raising the dose, and we have bigger and bigger impact on the tumor. And if we dose high enough, we can just completely obliterate the tumor in one dose. And that can have, but the challenge is in that case, because we're eliminating the tumor blood supply, all tissue within the tumor dies, whether it's normal tissue or cancer cells. And, so if you imagine you have a brain cancer and you just eliminate the tumor, that's very similar to surgery. So you mentioned chemical surgery, that at those very high doses, that's kind of what our drug does. It's like a surgeon, scooping out the tumor. And, but then, like in a brain cancer, that would be like having a stroke. That's what a stroke does, is kill part of your brain. And now it may be a good trade for cancer patients to trade their deadly tumor for a stroke that impairs them but doesn't kill them. But what we want to do is find the best clinical approach that not only gets rid of their cancer, but leaves them as well off as we can.
Yeah,
so,
it doesn't compromise their
yeah,
health otherwise. Yeah.
Yeah, so we want to keep as much function as we can and preserve as much normal tissue, normal cell function, but still get rid of the cancer. And so that's what, to a great extent, that's what we're searching for, is the right way to dose, the right way to treat, so that we bring the most benefit to patients.
Now, angiosarcoma, GBM, pancreatic, and liver cancers have high unmet needs. How are you prioritizing indications, sites, and potential combination partners without diluting focus?
Yeah, so that's a good question. So, so as I mentioned, we've chosen angiosarcoma as our quickest to approval indication, and that's for several reasons. One is that there's no approved drug for angiosarcoma, and so there's not much competition there. The off-label drugs that are used for it don't work very well. It's among the highest unmet need conditions, so the 5-year survival is under 12%, median overall survival about 13 months, and so it kills people pretty quickly, and that means it doesn't take that long to prove a survival benefit. You don't need a huge trial, so if you're working in 100% of patients and extending their lifespan indefinitely, then you don't need a lot of patients to prove you're beating alternatives in which people die in a year. So so that's an attractive indication. And so the special thing about angiosarcoma, the cancer is of endothelial cell origin, and endothelial cells are innately high expressors. So potentially 100%, every single angiosarcoma cell is sensitive to our drug. Whereas in other cancers, it's only a subset of cancer cells, the more malignant cancer cells, that are sensitive. So we think we can have a very big impact there. And another benefit is it's a rare cancer, which has some regulatory benefits, but it also means that you can get the manufacturing set up quicker. So if you don't need to produce very large amounts of drug, you can have a smaller scale supply chain, and we can get that set up more quickly. So we can get to approval faster. The situations you mentioned, glioblastoma, pancreatic cancer, liver cancer, bile duct cancers, cholangiocarcinoma, those are all great indications for us, and they have similar high unmet need to angiosarcoma. So they are very attractive indications for us. I would say those could be our four top priorities.
Yeah.
So there's kind of there's kind of several criteria that can be used to prioritize cancers in clinical development. So one of them is you need to get to approval in order to get revenue. So that's very important for a company. And so Angiosarcoma looks like our quickest path to revenue.
Okay, that makes sense. Yeah.
But then there's other cancers that are interesting for scientific reasons. So I mentioned, one of our goals is to find the way to dose and treat patients that preserves normal function as much as possible. Well, in order to study that, how to do that, you'd like to have a cancer where if you are damaging the normal tissue, you've got a clear ability to assess that. And, you can do quantitative measures of, how much has the normal tissue function come back. And I think things like brain cancers and glioblastoma are good for us in that regard because, you can, if you're harming the brain, you can usually tell, and so we can see cognitive neurological impacts, and so if you're treating your cancer for a stroke, how severe was the stroke, and can we find a way to dose that minimizes the severity of that stroke? And the things we learn in how to treat glioblastoma and preserve as much function, those may be applicable to other cancers as well. And cancers where we don't have as good feedback on how much normal tissue was damaged.
Yeah.
So a cancer like that may be important for scientific and medical reasons because they teach it's a good test bed for learning how to dose. It's not necessarily the easiest path to saving lives or generating revenue, but it may be the most valuable path to ultimately helping a lot of patients. And, clinical trials are scientific research. They're clinical research, and you're trying to learn. And, so sometimes you'll choose indications because they're educational. And they teach you things that you want to learn. And then there's other you always have to balance business and scientific and medical considerations. And, so it can be good to prove that you can successfully treat a large market cancer. So there's lots of judgment calls to be made, but
I think it's the quickest point to success, like the best.
Yeah,
best
it's
way
very
to-
important to have a quick
Yeah.
path to approval,
Right, especially when there's
because you can't. It's very expensive to help patients in the clinical trial setting, so it costs $150,000 or $200,000 to treat a patient in a clinical trial, and so to save 1,000 lives a year, you might have to spend $200 million, and it's not easy to get people to give you $200 million just
Right.
to help people without getting any revenue in return. So if you get to approval, then it can be profitable, and you can start treating thousands or even hundreds of thousands or millions of people. And so getting to approval is very, very important.
Now, manufacturing and CMC can make or break ADCs. What have you done to de-risk scale-up, stability, and quality for a nuclear-delivered construct?
Well, you have to work closely with manufacturing firms and the top firms in the industry. So, as a small biotech, so drug manufacturing, it's a very large scale activity. Most of these firms have hundreds or thousands or tens of thousands of employees, the firms that specialize in manufacturing. It's a very different mindset. Quality is very important. It needs a different type of person than, say, scientific research. And but they have a lot of expertise. So there's been many drug companies doing antibody drug conjugates, so they gained a lot of experience with other companies' drugs. And, so we have to, we engage those firms to help us with all of these questions. We really rely heavily on their expertise and their experience. But we have to work closely with them and make sure that they're doing what's right for our drug. And luckily, we have some very good partners in that regard. And then you have to work closely with the FDA and with regulatory experts who have experience working with the FDA and understand what the FDA wants out of the manufacturers and the quality assessments, the analytical assays. And so there's an ongoing conversation with the FDA, and so everybody has to work together and come to a common approach that ensures that the drug is serving patients well.
You raised significant capital, like you mentioned before, without the classic venture timeline pressures early on. What governance or cultural choices helped you keep patient-first engineering at the center?
Yeah, sometimes it's a challenge. You'd wish that everyone was motivated by saving lives and making money through saving lives. But sometimes people see their personal interest as a little bit different, especially when it takes 15 years to get a drug to approval. People may have their personal timelines that are a little shorter than that, and so I would say that's one of the entrepreneurial challenges in the drug development industry that makes it different than other industries where you can get revenue early on. And so if you can make a product that you can sell and get revenue, then your customers give you feedback, and the revenue helps everybody judge your company, and you can use that revenue to help bootstrap your company and build it steadily. Whereas in drug development, it's it definitely needs a great deal more faith that that the drug will make it to approval, will ultimately generate revenue, but
Yeah.
you don't get to sell it to customers and then ask the customers, oh, how was it? How was
Get
the
their feedback,
experience?
exactly.
Yeah, so so there's a long road. You have to do 15 years of work and spend $300 million before you have your first customer. And
Huge risk. Yeah.
so it's definitely a very different entrepreneurial challenge.
Yeah. Yeah. But I guess in the end, huge payoffs, not only financially, but results for the world. Yeah.
Yeah, that's right. Yeah, so you have to find the people who are motivated by
Yeah,
that.
by that mission, by that overall long-term goal
Yeah.
and mission. Oncology moves fast, and like you're saying, public hopes are high. How do you balance transparent storytelling with the ethical and regulatory constraints of a private company in an ongoing trial?
Yeah, that's another challenge. So, you both as a private company, you're regulated by the SEC, and you can't make a public offering of securities, so you can't just go out to the public and say, look, we need to raise money, we need to find investors to bring this drug forward. So you have to be careful what you say, and that does make it harder to fundraise. So so that's one challenge, and then the FDA similarly regulates what claims you can make about drugs, and you can't claim efficacy or safety ahead of approval and regulatory validation. So it's definitely living within those regulatory limits poses some challenges. It, so but fortunately, you can generate a lot of data, so both in animals and then when you're in the clinic in patients. So if you traverse the right steps and build relationships with people and talk to them within the context of those relationships, and often under confidentiality agreements and share the data, then it becomes possible to to tell your full story. And so I would say there are definitely challenges in this industry, but we've been fortunate. We've been able to overcome them. So the challenges haven't derailed us.
Yeah, no, and you're getting close to the end here. Yeah. So if AGX 101 succeeds, what could standard cancer care look like 5 to 10 years from now? And where does Angiex go next? New payloads, combo regimens, earlier stage disease? What does the future hold?
Yeah, so there's a lot of things we have to learn in the clinic, and so among those will be, when is the patient best served by combination therapies? When are they best served by taking our drug alone? Definitely the best-selling drugs today are immuno-oncology agents that help the immune system attack the cancers. And, so combinations with those drugs will be very important for us and very valuable for many patients. So, that's definitely an area we have to learn about. And, so there's a lot to learn about how to bring the greatest possible benefit to patients. And, so that's what we spend a lot of time thinking about and trying to generate a clinical research strategy that, helps us get those answers as quickly and efficiently as we can.
Right. Yeah. No, exciting times and a lot of promise, and I'm very excited to see what this new technology brings. And so, Paul, thank you for taking us from star-making physics to tumor-targeting biology. This has been a fascinating conversation. For listeners who want to connect with you or learn more about your work, what's the best way to reach you?
I would say go to our website, angiex.com. There's a section for patients, so if you'd like to consider enrolling in a clinical trial, there's there's a Q&A page for people considering that, which has guidance on how you would do that. There's also some contact emails, and there's a form on the website you can use to contact us. So we definitely welcome contacts from anyone who thinks they may be able to help us or who thinks we may be able to help them. But even if you don't want to contact us, I would say, keep an eye out. And as you hear things about us, if we have good news and that we're able to publicize, you can share on social media and, help more people learn about us. And I think, if we really have found a way to help a lot of cancer patients, then it will be good if people hear about it.
Yeah.
We've been pretty stealth as a company, so not many people know about us. But it's time for us to stop being stealthy and spread the word
Spread the
about.
word. Yeah,
And
absolutely.
so, yeah, and thank you for having me on today so that we can
Yeah.
do
No, I will
better.
certainly be following along. This is very, very exciting, breakthrough in medical technology and revolutionary. And so I'm, very, very excited to see what comes.
All right,
All
thank
right.
you.
Well, Paul, yeah, thank you again. And to everyone tuning in, don't forget to follow and subscribe so you never miss an episode of Industry Ignited. And until next time, stay bold, stay curious, and keep igniting industry.